Good morning. Good morning, everyone. Sorry for this late start. Yes, thank you for typing your name into chat for me. Yes, I apologize. I apologize for the late start today. I got up to no wireless. And let me say, I don't understand wireless technology in the least. I don't. But I have learned that when this particular problem happens for me, which is supposedly my Wi-Fi network is working, but I'm not connected to the internet. Yeah, I love that. I'm like, okay, that doesn't make any sense to me mentally. I have learned that I have to clear the cache in my router, in my combination modem router. I don't understand it. I don't know why that works, but there is this long process that you could do on a computer that will clear the cache and sometimes we'll bring it back. So here we are. Here we are, my dears. All right. Valerie, if you can hear me. Valerie, if you can hear me, great. If you can't, try exiting and coming back. I'm just going to type this out just in case she still can't hear us. Yeah, if you ever come into a Zoom meeting and your audio's not right, obviously try clicking the audio on and off a couple times. But if that doesn't work, sometimes it will help to leave the Zoom meeting and then come back into the Zoom meeting. Again, I don't know why that works, but it does. So, oh my goodness. All right, everyone. So, welcome to a new week. Happy Monday. It's going to be a very hot day today, so be very careful if you're outside for any reason, if you have to do any work outside, or if you're working in an unair-conditioned place. Please take good care of yourself. Lots of breaks, lots of water. Okay, I went out to water my garden at 6 o'clock this morning and it was already hot. So be very careful. Take good care of yourselves. All right, so today we only have one housekeeping announcement, and that is that you have your next lecture exam on Wednesday. So this lecture exam, just like the other ones you've had, will take place during the day on Wednesday from 7 o'clock onward. You just need to make sure you get the exam completed and submitted by 8 p.m. The exam will cover the material that we've gone through since the last lecture exam. The exam will be approximately the same length and approximately the same types of questions. I will tell you that there are, I think, 4 short answer questions on this exam. So the rest of them are multiple choice. And there will be a bonus question, as usual, on the lecture exam. I got your message, Lacey, no problem. Okay, we will probably have to go past 11 o'clock this morning since we got a late start. If you need to exit the meeting at 11 to go on about your day, that's not a problem. Feel free. You'll just want to take a look at the lab video when you have a moment and watch the last, whatever, 20 minutes of it, whatever you have to miss. Okay. And our subject today for lab is oxygen. Since we're starting to talk about metabolism, we're going to start off with a laboratory testing technique, which looks to put organisms into what we call oxygen categories. And we're going to be talking about a very particular type of media that is called Thioglycolate media. And you don't have to write that down yet. You'll see the word in just a minute. This is a semi-solid type of media. It comes in a tube, in a test tube. And by growing cells in this media and looking at the pattern of how the cells grow through the tube, we can assign that microbe, that bacterium, to one of 5 different categories of how they relate to oxygen. Is oxygen a requirement for them? Is oxygen toxic to them? Or do they fall somewhere in between? So that's what we'll be looking at today, is how oxygen relates to the survival and growth of bacteria in culture. Again, we want to know these things about bacteria. We want to use something like thioglycolate culture media so that we can learn something about a microbe, so that we can identify that microbe. So that's why we would put an unknown sample of cells into a thioglycolate tube. We would want to see how those cells grew so that we could determine what their oxygen category is. And that gives us a piece of information about them and helps us identify them. All right. I'm going to go ahead and pull up our slides. And that's what you should be seeing on your screen now. The title slide that says Oxygen and Growth. So we have a long list of objectives today, 5 different things. But first and foremost, we're going to talk about oxygen categories. We're going to talk about obligate aerobes, what their needs are, obligate anaerobes, what are called aero-tolerant anaerobes, micro aerophiles, and facultative anaerobes. You may have heard of some of these categories, but some of them may be new to you. And again, the reason there are 5 different oxygen categories for bacteria is because bacteria are incredibly diverse when it comes to their metabolic abilities. And that includes a lot of diversity in their relationship to oxygen. We'll talk a little bit about this medium, this thioglycolate medium. And we'll talk about the way that bacteria grow in it. Again, the patterns of growth. We are literally going to be able to look at the cells in the tube in how they're growing, what shape they have assumed within the tube. And that's going to allow us to assign them to one of the categories. We're going to talk just a little bit about specific culture methods that we use when we're dealing with anaerobic organisms. And moving away from oxygen and oxygen categories, we're going to talk about what's called the Catalase Test. And then we're going to run a little experiment, just like we did last time with the MSA and the EMB agar. We're going to run our own little experiment. We're going to take 4 different bacteria. We're going to inoculate thioglycolate medium and then incubate the tubes and evaluate the growth patterns in those tubes to see if we can figure out which organism is which. So I always like to start off this oxygen lab with a question. And that question is, why do human beings need oxygen? You can talk to most high school students, probably middle school students, even elementary school students, and they can tell you that human beings must have oxygen. If they don't have oxygen, they will die. And of course, those of us in healthcare related fields, we know that in an emergency situation, when we're faced with helping a person who is flat out on the floor, unconscious, you know, we talk about the ABCs, right, in emergency medicine, and the A stands for airway, because that very first thing you got to think about is oxygen. Is this patient getting oxygen? Why? So Lacey's saying, well, you have to have oxygen because your tissues have to have oxygen. Your tissues have to be perfused with oxygen. And that's right. That's right, Lacey. We breathe the oxygen into the lung, and then there's a gas exchange event, right? Between the alveoli, those grape-like clusters in the lung, and the red blood cells. Carbon dioxide is released to be exhaled, oxygen is taken in, and then it travels throughout the body and it exchanges with the tissue. So every cell in the human body gets oxygenated constantly. Why? Why? Why do your cells need to be oxygenated? Why do your tissues need to be perfused? Why? What's happening with that oxygen? Any idea? It is about metabolism. You better believe it. It's about metabolism. Oxygen is involved in a very important metabolic pathway in human beings. A metabolic pathway that is so important, in fact, that if you go without oxygen for minutes of time, you will die. Minutes of time. That's it. You're dead. What's that oxygen doing? Lacey's got the pathway, cellular respiration. Cellular respiration. And let me change the question now. Let me change the question. What is the product of cellular respiration? And more importantly, because cellular respiration is connected to what's called oxidative phosphorylation in humans. What's the end product? So you go all the way through cellular respiration, you go through oxidative phosphorylation to make, anybody know? What has to be made in every single cell in your body, 24 hours a day, 7 days a week, through your entire life? What molecule is so important to your survival that you can't go more than a few minutes without it? Carrie's got it. Lacey's saying glucose. Lacey, glucose goes into cellular respiration. Glucose is the starting molecule, the fuel for cellular respiration in humans. That's right. But what's coming out of cellular respiration, again, and oxidative phosphorylation, is ATP, adenosine triphosphate. This molecule is essential to life. Every cell on the planet requires ATP. Bacteria need it, archaea need it, eukarya need it. Animals need it, plants need it, fungi need it. ATP, single most important molecule in a cell. And you probably learned at some point in your education that ATP is sort of this energy molecule. It's got something to do with energy. It's made your mitochondria. And we always teach students that mitochondria are these little powerhouses. They make energy. What they're really making is ATP. And cells are using ATP as a means of conserving energy. So as we start talking about cellular respiration, really from now till the end of the course, understand that it's all about ATP. Fermentation, another metabolic pathway that we're going to talk about because it's very common in bacteria. Fermentation is also about making ATP. ATP is the central player in these metabolic pathways. That's how important it is. The unfortunate thing from an evolutionary standpoint is that your body, your human body, does not store ATP very well. Your body makes ATP as it is needed. You have about 4 or 5 minutes worth of ATP in your cells at any one time. And that means if you stop breathing and you fall down because you are not breathing, you're on the clock towards death. If we cannot get oxygen back into your bloodstream so that it can be carried to your cells, you will die. And it's because you cannot make new ATP. You've got just a little window of it available at any one time, and you can't live without it. Now, my original question about oxygen was, what are your cells doing with oxygen? And I'll tell you the answer to that, but it might not make much sense to you right now until you've gone through this lecture about cellular respiration. But at the end of cellular respiration, before oxidative phosphorylation occurs, the glucose molecule that Lacey mentioned, has been completely broken apart. And all of the electrons in all of the bonds in glucose have been released. We haven't talked about it yet, but those electrons can't be left to their own devices in a cell. When things lose electrons, and we call that process oxidation, when things lose electrons, other things have to take those electrons on. And we call that process reduction. So glucose is going to be oxidized during cellular respiration. All of its bonds are going to be broken apart, and all of its electrons are going to be released. Something else has to get reduced. Something else has to receive those electrons, gain those electrons. And at the end of cellular respiration, at the long end of all these metabolic reactions, because there are many of them, at the end, it's oxygen that does that. Oxygen is what we call the final electron acceptor at the end of cellular respiration. Oxygen receives all those electrons. It combines with a little bit of hydrogen, and it makes water. If you have no oxygen available, cellular respiration stops. And if cellular respiration stops, ATP production stops. That's what the oxygen's doing. The oxygen is receiving the electrons that were set loose from glucose. It's receiving them and it's converting into water. And that's why you need oxygen. All right. So back into our lecture slides. This is a drawing of the ATP molecule right up here at the top of the slide. It's a pretty small molecule. It's pretty basic. Again, it's called adenosine triphosphate. Tri, of course, means 3. There are 3 phosphate groups on this particular molecule. The adenosine is this part of the molecule right here. This ring structure and this nitrogen-rich structure that's attached to it. This is the adenosine part. And then over here, sticking off to the left, these are 3 phosphate groups. Phosphate groups are phosphorus and oxygen combined together. There's one here, there's one here, and there's one here. ATP. And I told you a minute ago that ATP is used sort of like currency in a cell. When we break this last phosphate group off of the molecule, a little bit of energy gets released. Remember, when you break a bond, electrons get released, and electrons are sort of the couriers, if you will, of energy in a cell. So you break a bond, you release electrons, because that's all the bond is, right? A pair of shared electrons between 2 atoms. When you break the bond, you release the electrons, you release energy. And that energy can be used by the cell to do certain metabolic reactions. Now, we're going to be focused on 3 different reactions in this course, and we're going to talk them to death. You're going to be so sick of them, you'll probably be dreaming about them by the end of the course. One of them is aerobic cellular respiration. One of them is anaerobic cellular respiration. Those are 2 different things. And the last one is fermentation, as I mentioned earlier. Human beings only use this first one when conditions are good, when everything's normal, when our cells are just metabolizing away. We are capable of number 3 here. Human beings can ferment under certain conditions, but it's considered a secondary pathway, one that we don't do for very long, one that is not preferred. Our cells prefer to do aerobic cellular respiration in order to make ATP. Microbes can do any and all of these. There are some microbes that are capable of all 3 of these pathways. And what's really shocking is that there are some microbes that do all 3 of them equally well. In other words, no pathway is preferred. Microbes are very diverse. Some of them only do aerobic respiration. Some of them only do anaerobic. There are some microbes that only ferment. And there are some that can do all 3 or can do some combination of 2 of those. So the reason we focus on these 3 pathways is because we see all 3 of them so often in bacteria and other microbes. And we have to have a way to test. We have several ways to test to see what they're doing. What kind of metabolism are they doing in order to make their ATP? So we can grow microbes in culture to test them, to figure out what their oxygen requirements are, what category they belong to, and what metabolic pathways they use. And we can adjust culture conditions. As we've talked about in here, we can add ingredients to culture. We can take ingredients away in order to sort of push the microbe to use one or another of those metabolic pathways. All right, the next thing we're going to do is talk through those 5 different, what we call oxygen categories. Remember, we said obligate aerobes, obligate anaerobes, aero-tolerant anaerobes, micro aerophiles, and facultative anaerobes. We'll start with the easiest ones, the obligates. That word kind of gives it away, doesn't it? An obligate aerobe is an aerobe that, or I should say, is a microbe that requires oxygen in order to make ATP. You and I are obligate aerobes. We must have oxygen or we won't survive. And there are some bacteria that fall into this category, obligate aerobes. These are bacteria and other microbes that are using aerobic cellular respiration. And sometimes they can also do fermentation as a secondary pathway. In other words, if for some reason they temporarily don't have enough oxygen, they can switch over to fermentation for a bit, but then they're going to want to switch back as soon as possible. This all varies according to the species of bacteria. Like I said, there are some bacteria that can do fermentation for long periods of time and not suffer any consequences. Now, obligate aerobes, let's say bacteria that are obligate aerobes, have an enzyme inside of them called catalase. And we're going to talk about this enzyme. Catalase is an important enzyme in any organism that is routinely using oxygen. And that's because oxygen, believe it or not, can be quite toxic to a cell. Yes, we need oxygen. You and I, we need oxygen, and some bacteria as well. But anytime oxygen is part of a metabolic pathway, anytime there's lots of oxygen within a cellular environment, there are byproduct molecules of oxygen that can be made, that get made. One of these is called hydrogen peroxide. You probably have some hydrogen peroxide in your medicine cabinet right now. It makes a great disinfectant, doesn't it? Or I should say antiseptic. You can put it on little cuts and scrapes and it'll help clean them out. Hydrogen peroxide is toxic. It's toxic. So cells that might have hydrogen peroxide around, because they tend to have oxygen around, those cells also need a way to get rid of the hydrogen peroxide. And that's what the catalase enzyme does. You and I keep our catalase enzyme under lock and key. In our cells, we keep our catalase enzyme in one of our organelles. Does anybody remember which organelle in human cells is responsible for breaking down hydrogen peroxide as one of its jobs? Which organelle in a human cell is responsible for getting rid of hydrogen peroxide? Does anybody remember? Good. Carrie's got it. Lacey's got it. Marilyn's got it. Peroxisomes. It's in the name a little bit, right? Peroxisomes. Marilyn, it's not the lysosome. It's the peroxisome. Peroxisomes have a couple of jobs, but one of their most important jobs is breaking down hydrogen peroxide. And they do that using this catalase enzyme. Now, if you're an organism that needs oxygen, that means lots of oxygen is coming into you all the time. And oxygen can be toxic. There are dangerous byproducts of oxygen that can be made. One of them is hydrogen peroxide. Another one is called oxygen radicals or free radicals. You've probably heard of antioxidants, foods and nutritional supplements whose job it is to help fight against these radical molecules. And one of the most common radicals that we see in humans is an oxygen radical. That term radical in chemistry just means that it's an atom that has an unpaired electron in its valence shell. And as we've talked about, any atom that has an unpaired electron is unstable. And oxygen radicals are unstable. They don't like having an unpaired electron in their valence shell. So they go and they try to steal an electron from somewhere else. And that stealing causes damage. And that's why radicals are so bad for us. They age our cells and they can bring on diseases. So oxygen is not a benign molecule. Yes, you have to have it as a human. And yes, there are bacteria that have to have it. But it can also form dangerous byproducts that are toxic to cells. One of the things that you learn in medicine is that while we often give patients 100% oxygen through a mask or a cannula in emergency situations, as soon as possible, we get them back on room air, don't we? It's not good to be on 100% oxygen for long periods of time. And that's because of these dangerous oxygen byproducts. So as soon as we can, we wean a patient off of 100% oxygen, get them back on room air. Our cells can deal with a small amount of oxygen. Does anybody know how much oxygen is in room air? What's the percentage? Anybody know? A lot of people, lay people, people that are not science students, they think that air is oxygen, right? But actually, it's mostly nitrogen. It's mostly nitrogen. How much oxygen is there in air? A little bit more than that, Carrie. Carrie's saying 8-ish. It's more than that, but it's not a lot, really. It's about 21%. Room air has about 21% oxygen in it. So yeah, there's oxygen. You have to have oxygen or you wouldn't survive, but it's not all oxygen. Yeah, around 20%, 21%. Good. Okay. The opposite, basically, of an obligate aerobe is an obligate anaerobe. An obligate anaerobe finds oxygen toxic. If those organisms get exposed to oxygen, the oxygen will kill them. So they aren't using oxygen to make ATP. They're doing a completely different metabolic pathway to make their ATP. They are using anaerobic cellular respiration and or fermentation. It varies by species again. So these are organisms that they don't use oxygen as their final electron acceptor at the end of cellular respiration. They use a different molecule. They still have to have a final electron acceptor. Something has to receive or gain the electrons that got set loose when glucose was broken down. But they don't use oxygen for that purpose. They use something else. Now, these organisms, because they don't require oxygen, they don't have oxygen floating around inside of their cells because oxygen is toxic to them. So generally speaking, you won't find the catalase enzyme inside of these kinds of bacteria. They don't need it. If there's no oxygen around, there's not going to be any hydrogen peroxide around. And if there's no hydrogen peroxide that needs to be broken down, you don't need to make catalase. Now, the third category are the aero-tolerant anaerobes. These are organisms that don't use oxygen to make their ATP, but the important distinction is it's not toxic to them. So it's okay for an aero-tolerant anaerobe to be in the presence of oxygen. It's not going to kill them. But they're not going to use it. They don't use oxygen as their final electron acceptor. They use something else. Similar to the obligate anaerobes, the aero-tolerants don't typically have catalase. I also have an interesting little note down here, more of a trivia note than anything. Aero-tolerants also don't have a problem with hydrogen peroxide because they don't have the system inside their cells that generates hydrogen peroxide. So hydrogen peroxide is not a problem for them because their cells don't make it from oxygen. So they don't need catalase either. If there's no H2O2 around, you don't need to break it down. So you don't need the enzyme that would break it down. Aero-tolerant anaerobes typically use fermentation as their primary metabolic pathway for making ATP. But, and this is important, put a star next to this in your notes, they grow best in the laboratory in conditions where there's a little bit of oxygen around, low oxygen concentrations. They are not using that oxygen to make ATP, but they grow best when there's just a little bit in the air that surrounds them. And I can't tell you why because I don't know why. So they're not going to use that oxygen, but for whatever reason in physical chemistry or atmospheric chemistry, they like to have a little oxygen in the air around them. They grow best that way. Again, these are things that are helpful for us to know when we're trying to grow these organisms in the laboratory. All right, let's take a look at what these organisms look like when they're growing in this special culture media called thioglycolate broth. I told you that thioglycolate is a semi-solid culture medium. So this is the first semi-solid one we've seen. Basically, what you do is you take a basic basal media. Remember, we said a basal medium is a chemically defined medium. You take that and you add just a little bit of agar to it. Not enough agar to turn it into a solid, just enough to thicken it up a little bit. I like to think of it as the way you might add a little cornstarch to a recipe to thicken it up, or you might add a little flour roux to a recipe if anybody's a cook. You might add that just to thicken up, say, a stew or a soup. We're adding just enough agar to thicken this broth up just a little bit. And there's a reason we do that. When you put a culture medium into a test tube like this, the oxygen in the air, when the cap is off anyway, the oxygen is going to dissolve into this liquid, but it's only able to dissolve to a certain point. When we add agar, just a little bit of agar to this liquid, we're going to prevent the oxygen from penetrating into this medium very far. It's only going to be able to penetrate into about the top 20%, the top 1/5 of the tube. So if I were able to measure the oxygen concentration, I would find oxygen up at the top region of the tube. But as I kept going down, down, down into the medium, there'd be less and less and less oxygen. And at the very bottom, there's no oxygen. So what I've done in my thioglycolate broth is I've created a gradient of oxygen. And that's going to allow me to see, again, the pattern of growth. Where will the cells grow inside this medium? Will they grow where there's lots of oxygen? Will they grow where there's no oxygen? Or will they grow somewhere in between? So this tube, this drawing over on the left-hand side here, this represents the kind of pattern that we would see in a thioglycolate tube if we were growing an obligate aerobe. The little red dots represent the cells, the bacterial cells. And as you can see, they're all clustered up in the top of the tube. So I'm going to see growth. I'm going to see that, remember, when we see cells growing in a broth, it becomes very cloudy, very opaque looking. I'm going to see growth, but I'm only going to see it up at the top. I'm only going to see it in that top 1/5, that top 20%, where the oxygen is. Because obligate aerobes must have oxygen or they won't survive. Now compare that to this tube. If I'm trying to grow an obligate anaerobe, the only place that those cells will grow is all the way down at the bottom. They're not going to grow at the top because there's oxygen there, and oxygen is toxic to them. They're not going to grow down in the middle of the tube either, because even though there's less oxygen in the middle of the tube, there's still oxygen, and oxygen is toxic to them. So they're only going to grow down at the bottom. Now, I have a little note down here underneath tube B that says, cells often clump together in thioglycolate medium, and they grow as these little tendrils because of that agar that we put in the broth. Now, you can't see any tendrils in these drawings, but you'll see them when we look at actual images of thioglycolate tubes. So just keep this as a note in the back of your mind. Now, the other category that we talked about so far are the aero-tolerant anaerobes. And I have to apologize to you because the artist that drew this didn't do as good a job drawing this tube as I would have liked. Remember, we said aero-tolerants don't use oxygen to make their ATP, but oxygen is not toxic to them. So you would expect aero-tolerants to be able to grow anywhere in this tube. They don't need oxygen, so they can grow where there is oxygen, where there isn't oxygen, and anywhere in between, right? And that's basically the way the artist drew the tube. But remember, the other thing we said about aero-tolerants is that they grow best in low oxygen concentrations. So in this tube, there's a place where you're going to have lots of, you're going to have oxygen, but you're going to have it at a low concentration. And it's essentially the bottom half of this oxic zone, this top 20%. They're not going to grow right up at the top of this zone. They're going to grow in the lower half of this zone. That's why I've drawn this red bar right here. When you grow aero-tolerants in the lab, you're not going to see growth at the very top of the tube. And you're going to see most growth in the lower half of this oxygen-rich zone, this oxic zone. That's where you're going to see the most growth. That's what the red bar is. You'll also see growth through the rest of the tube. You're just not going to see as much as you will see right here in the lower half of the oxic zone. So again, the reason I drew the bars is because the artist didn't quite get that across. We're going to see a lot of growth here, and then we're going to see those weird tendrils of growth sort of hanging down through the rest of the tube. And again, I'm going to show you some actual thioglycolate tubes when we talk through our experiment. Now, the fourth category of oxygen requirements is the micro aerophile. Now, if you break this word down, micro, of course, can mean small amounts. Aero refers to oxygen, and phile means love or like. So these organisms like a small amount of oxygen, micro aerophile. Micro aerophiles require oxygen to make their ATP. So in that way, they're similar to the obligate aerobes. But the difference is micro aerophiles need a reduced concentration. Now, as another note, they grow best in a little bit more carbon dioxide than we normally see. 5 to 10% carbon dioxide and that low oxygen concentration. One of the nice things about modern microbiology labs and modern incubators is that not only can we control the temperature inside an incubator, we can also control the carbon dioxide level. When you close the door on an incubator, there's a tight seal that's formed. In fact, most incubators have 2 doors that close. That way, you can add a little more carbon dioxide gas to the incubator and bring the concentration up. Normally, there's about 5%. So if we didn't do anything, there would be about 5% carbon dioxide in that room air. But these organisms like just a little bit more than that. They grow best when there's more like 5 to 10%. So just another little thing that is helpful to know if you're trying to grow an organism that happens to be a micro aerophile. If you bump up the carbon dioxide in your incubator, they'll grow better for you. Okay. The other tool that we have that we used to use more than we do now is called a CO2 jar. Like I said, modern incubators allow you to change your CO2 level in the incubator with ease. But we used to use what's called a CO2 jar. And basically, this was just a jar, with a tight fitting lid. And what you would do is you would put your culture container, after you had inoculated it, you would put it into the jar. You would light a candle and put that in, and then you would put the lid on. Along with the tightly fitting lid, what's going to happen is that candle is going to burn off some of the oxygen in the air. So the candle will reduce the oxygen. Remember, it's a micro aerophile. It likes lower oxygen levels. And you could get these micro aerophiles to grow for you by using this jar. You can even purchase, if you still have a CO2 jar in your lab, you can even purchase now a CO2 generating packet. You can buy these little packets. They look like juice boxes. You take the lid off the jar and you put your culture container in. You poke a hole in this little juice box package and you put it in. and you put the lid on. You don't need the candle. What's going to happen is that little package is going to release carbon dioxide. It'll drop the oxygen levels and it will bump up the CO2. It's an easy way to do it if you don't have one of these modern, expensive incubators. But I will tell you that in any modern diagnostic laboratory that you would find, like in a hospital, they're going to have a modern incubator. So CO2 jars are kind of a thing of the past now. But it used to be a way that we could grow micro aerophiles in the lab. All right. The last oxygen category is the facultative anaerobes. And you'll notice I have little asterisks next to this category because most bacteria fall into this category. Most of the bacteria are facultative anaerobes. These are organisms that are going to grow best in the presence of oxygen, but they don't require oxygen. And that's because these are those organisms that can switch back and forth between metabolic pathways depending on the conditions that they find themselves in. So if there's oxygen available, they'll do aerobic cellular respiration. If there's no oxygen available, well, they might switch to anaerobic respiration, or they might switch to fermentation because they can do all of them. So if you think about it, if you had to choose an oxygen category, this might be the one you would choose, because a facultative anaerobe is a very adaptive kind of an organism. It's not going to die just because there's no oxygen around suddenly. If it finds itself in an environment where there's no oxygen, it's going to be just fine because it can switch back and forth between metabolic pathways. Pretty good trick if you think about it. If we go back to our drawings, here is the picture of our micro aerophile. Again, it's a lot like tube A. It's a lot like the obligate aerobe in that you're only going to see growth up in that top 20%, up in the oxic zone or the oxygen-rich zone. But notice, it doesn't grow all the way to the top of the liquid. It doesn't grow all the way up to the top of the tube. It grows in the lower half of the oxic zone. It likes a little less oxygen than you would find right up at the air to liquid interface. Micro aerophiles. And finally, we have our facultative anaerobes, the tube right in the middle. This tube looks very similar to the Aerotolerants with one big difference. Remember, the Aerotolerant is going to grow best in the lower half of the oxygen-rich zone, but it will grow through the rest of the tube as well. The facultative anaerobe is going to grow all the way through the oxic zone. In fact, it's going to grow best in the oxic zone. That's why you see the heaviest red right here. It's going to grow best right there at that top of the tube, but it will also grow through the rest of the tube. It doesn't require oxygen, but if there's oxygen around, it will use it. It prefers to do aerobic metabolism, but if there's no oxygen around, it'll just switch to another pathway. So 5 categories, 5 different patterns of growth. And by examining a culture tube that's been in the incubator for 24 hours, we can assign a microbe to one of these categories. Now, I mentioned the Catalase test, so this is a good place for us to talk about that. It's a very simple test. It's one of my favorite tests in the lab because it's very fast to do, very inexpensive, and you get immediate results. Remember, the Catalase test is looking for the presence of the catalase enzyme. So all you have to do for this test is expose the bacterial cells to some hydrogen peroxide. If they have catalase, they're going to start breaking it down. If they don't, they won't. Now I have the reaction here for how hydrogen peroxide gets broken down, because this is important. When catalase is around, it's going to break apart hydrogen peroxide, that's H2O2, into water and oxygen gas. So if catalase is present, you're going to see oxygen gas being released in the form of bubbles. And that's what you can see over here in the image. When you're ready to do a catalase test, what you do is you take a little bit of culture material, maybe pick up a colony off of an agar plate, or take a loop full of broth culture and you put it onto a microscope slide in the center of a microscope slide, like you were making a smear to examine under the microscope. And then all you do is you place a drop of hydrogen peroxide on top of that smear of cells, and you watch. If there's catalase in those cells, this is going to happen. It's going to immediately foam up. And that's because those cells are using the catalase to break down hydrogen peroxide, and you're going to get oxygen released, oxygen gas. And it's going to bubble up just like this, and it happens immediately. If there's no catalase enzyme in the cells, this is going to happen. It's just going to sit there. You can see there's a big drop of hydrogen peroxide sitting on top of this smear, and nothing is happening. Okay. Now, remember, in human cells, the catalase enzyme is found in an organelle. It's found in the peroxisome. But in bacterial cells, there are no organelles. Remember, that's what makes a prokaryote different from a eukaryote. There are no organelles. So that means the catalase enzyme is just in the cytoplasm in a bacterial cell. So as soon as you expose that cell to hydrogen peroxide, the hydrogen peroxide is going to come in. The catalase is just immediately going to start breaking it down. And you're going to get the bubbles. Easy, easy test to do and very inexpensive. All you need is a bottle of hydrogen peroxide from the drugstore. Now, one of the things on our objective list for today was talking just a little bit about growing anaerobes. Look, it can be hard to grow anything other than an oxygen-using microbe, because if you have a microbe where oxygen is damaging to them or will kill them, you've got a problem. As soon as those cells get exposed to room air, you've got a problem with dying and dead cells. So it can be hard. It can be hard to grow an anaerobe successfully. And I'm talking about an obligate anaerobe primarily, an organism where oxygen is toxic to it. So if you want to grow an anaerobe, you've got to get all of the oxygen out of its environment, and that can be a little tricky. There are two ways that are commonly used, and one that's, or I should say two that are uncommonly used. I want us to be familiar more with the common ways that we can get rid of oxygen and produce an environment where an anaerobic organism can grow. So there's something called a Brewers jar, and more specifically, a Brewer's Anaerobic jar. And this jar was named after someone, which is why we use a capital B for the word Brewers. Similar to that CO2 jar I mentioned, this is a literal jar. I'll show you a picture of it in a minute. And it comes with these packets, these packages that, again, they kind of look like juice boxes. One of them is a hydrogen gas pack. And then we also get an oxygen indicator pack. So essentially what you do, just like we talked about with the CO2 jar, you take your culture container, whether it's an agar plate or maybe a broth tube. You take your culture container, you inoculate it, you put it into the jar, and then you open up these packs and put them in, and then close the jar tightly. The hydrogen gas pack is going to release hydrogen gas, and that hydrogen gas is going to combine with all of the oxygen that's in the air inside that jar. And hydrogen gas plus oxygen just makes water. After you put the cap on to these jars and screw it down tightly, if you watch the jar for a few minutes, you'll start to see condensation forming on the inside of the jar, and that's this water. As the oxygen is being removed, water gets formed. Now, I mentioned this second pack. This is an oxygen indicator pack. This pack is there to tell us if any oxygen accidentally gets into that jar while the jar is incubating. So the only reason this pack is in there is because if any oxygen accidentally gets into the jar, maybe there's a loose seal on the jar, for example, this indicator pack is going to change color. It's called an indicator pack because it's got indicator dye in it. And if any oxygen accidentally gets in, that pack is going to change color. After we put everything in the jar and we tighten down the lid, we're just going to put that whole jar into the incubator. And we're going to leave it just like we would any other culture. And in the morning, we'll come back, we'll take the jar out, and through the walls of the jar, we'll be able to see if we had any growth overnight in our culture container. So that's a brewer's jar. The other common way that we create an oxygen-free environment is with our thioglycolate tubes, the ones we just talked about. Remember, at the very bottom of a thioglycolate broth tube, there's no oxygen down there. So an obligate anaerobe will grow at the very bottom of that tube. Now, I mentioned to you that thioglycolate broth is a basal culture medium with a little agar added. Well, one of the ingredients in that basal media is thioglycolate. Thioglycolate helps to get rid of oxygen. It actually combines with oxygen in the deep regions of that tube and gets rid of it. So it's the thioglycolate that's creating that zone with no oxygen down in the bottom of the tube. The only other thing that I'll mention about these thioglycolate tubes, because you might run into this someday in a diagnostic lab, most people buy their thioglycolate tubes. You know, we make a lot of our own culture media in the microbiology lab, but this is one that we don't generally make ourselves. We buy it pre-made. And that's because you need to make this, you know, you need to be very careful when you're making this media in order to get the right oxygen gradient. You can buy thioglycolate tubes in two forms. You can buy just the regular ones, which are the ones I've been talking about. But you can also buy a thioglycolate tube that has a little bit of an indicator dye in it. And just like the dye in the Brewers jar, this dye is going to change color if it gets exposed to oxygen. Remember, the very top 20% of a thioglycolate tube is going to have oxygen in it. So this dye will turn the top of the tube a reddish pink color. In other words, you can buy a thioglycolate tube that shows you exactly where the oxic zone is. You can buy a type of thioglycolate tube where you can see exactly where the oxic zone is. Most people that I've worked with don't bother. They just buy regular ones because the ones with the dye in them are just a little bit more expensive. And you can judge where the top 20% is pretty easily. You don't have to have an indicator dye, but you should know that you can buy those tubes with an indicator dye. So there's no guessing involved whether or not cells are growing in that oxygen-rich zone at the top of the tube. Okay, so I mentioned that there were two commonly used things to create an oxygen-free environment. There are two that are less commonly used, and I have those listed down here. You can buy an anaerobic incubator. You can buy an incubator that will remove every trace of oxygen from inside. As you might imagine, these are very expensive incubators. So generally speaking, you only would find one of these incubators in a place where they are working with a lot of anaerobes, generally a research laboratory, or I'll say a very large hospital system might have an anaerobic incubator. But most places that I've been in, that I've worked in, do not have these. They're just too expensive. And finally, there's also what's called an anaerobic glove box. And that's the picture I have for you over here. This is not an incubator. There's no way to heat up the air in here. It's not going to create a nice dark space in here like an incubator would. But it will create an oxygen-free zone. And that's because it's got a valve here that you can draw the oxygen out and pump in carbon dioxide. You'll notice it's a glove box. You can put your hands in here through these gloves that are attached to the wall. And you can see you can open and close this thing with this sealed door. So you can get your culture containers in and out. So this is just called a glove box, and it's an anaerobic glove box. Again, not common. I don't see these, in my experience, in a lot of laboratories. On this slide, you're looking at a picture of a brewer's jar. That's what this is. These are just made out of a hard plastic material. So you can see into them, you can look at your culture containers, you can check on them, you can see if anything's growing. It's a hard plastic container. It's probably about a liter's worth of space in here. So you could stack up maybe a dozen Petri dishes in here. Notice that the lid has this thumb screw on it. What you do is you screw the lid down by hand, and then you turn this thumb screw. You're going to create a really tight seal here. In this picture, you can see that we've added in these two packets, a silver packet and a white packet. You can just see a Petri dish at the bottom. The silver packet is the hydrogen gas packet, and the white one is the oxygen indicator packet. If this thing changes color, we get very sad because it means oxygen has accidentally leaked into the container and our cells are probably dead. Over here on the lower part of the slide, This is what thioglycolate medium looks like when you buy it commercially. Again, it's just a plain old basal medium, but it's had agar added into it. So if I were to tilt this tube back and forth, it wouldn't move the way a liquid moves. It's a little bit thicker than that. It's called a semi-solid. And finally, this picture over on the far left, the far right, this is a thioglycolate tube that has had an indicator dye added to it. See that? The indicator dye is turning pink where there's a high amount of oxygen up in the oxic zone. So without even growing any cells in here, I can already see where all the oxygen is. It just makes it easier when you're evaluating your tube after you've grown some cells in here. It just makes it easier to see exactly where the cells are growing, whether they're in the oxic zone or out of the oxic zone. Remember, there is a little bit of oxygen in the center of this tube, but there's no oxygen at the bottom. All right, lots of information. I know, lots of information. Does anybody have any questions? Anything that's occurring to you right now? Any questions about the 5 oxygen categories? And any questions about thioglycolate broth or a brewer's jar? Any questions coming to you right now? Just type them into chat and we'll talk about them, okay? What we're going to do now is switch over and do our little experiment. And we'll see if we can read or interpret the results in a thioglycolate tube. We're going to see if we can interpret a pattern of growth and determine what category certain microbes belong in. Okay? And you'll get a chance here to see what I mean when I say that cells tend to stick to each other in thioglycolate and they form these long strings, these tendrils inside the broth sometimes. Okay, so here are today's organisms. Here are today's bugs. We're going to be using a new organism called Clostridium Sporogenes. This is a gram-positive rod. We don't get to see a lot of gram-positive rods, but this is one. Here's a smear of Clostridium Sporogenes. I think you can see it has been gram stained, by the way, and we're under the 100x lens. And I think you can see that these cells have stained purple. These are gram positive, and they're also rod-shaped organisms. So gram positive rod. This organism lives in the human body. It lives in parts of our gastrointestinal tract. It lives in the tract of other mammals as well. But it's also found out in the world. You can find Clostridium sporogenes in soil samples. You can also find it in marine sediment. So if you were to go into the ocean, down onto the floor of the ocean and gather up some sediment, you would find Clostridium sporogenes there as well. So that's our first bug for the day. The second one you've seen before, Enterococcus Fecalis. You just saw this organism in our last experiment. This is a gram-positive coccus. Also can be found in the GI tract. It's enteric. One thing we didn't mention last time was that Enterococcus fecalis can be found in other parts of the body. We do sometimes find it in females in the vagina. Remember, the vagina is not a sterile place. And also in the mouth. But the primary place you're going to find E. fecalis is in the GI tract. Now, I put a little star next to the gram status of this organism. And I'll show you why. This is what E. fecalis looks like on a gram stain. E. fecalis is one of the organisms that we might call a coccobacillus. Remember we said there are cocci and there are bacilli or rods, but sometimes, rarely, you'll find an organism that's kind of halfway between those two shapes. And Enterococcus is one of those. Take a look right here, for example, or right next door, right here. There are two of the organisms right here, side by side. Notice that this organism is not really spherical. It really has more of an egg shape. Here, look at these two right here. You would be forgiven if you called this a coccus. It really looks much more like a coccus than a rod. But this is one of the few organisms that it's more egg-shaped than spherical. So some reference materials will call this organism a coccobacillus, halfway between a coccus and a rod. Now, the third organism today is our old friend E. coli. E. coli is a gram-negative rod, and it's an enteric coliform. It lives down in the intestinal tract, and it can ferment lactose, remember? So gram-negative, pink. Our last organism today has a very unusual name, at least pronunciation-wise. It's called Alkalogenese Fecalis. It's another Fecalis enteric organism. Alkalogenese, that's how you say that genus. This one is also a gram-negative rod, and it can be found in the GI tract. It's enteric, but it can also be found out in the environment, just like the Clostridium that we mentioned. It can be found in soil, and it can be found in certain types of water. So that's alkalogenese. Now, in today's experiment, I'm not going to take the step of growing the cultures on nutrient agar, but know that that was done, and all cultures were confirmed to be alive and well before we did the experiment. So I want you to just take a look at this list just very briefly and notice something. If I were to gram stain these four organisms, I would get some useful information. If I got these organisms into the lab and I didn't know what they were, I would get some useful information by gram staining them, wouldn't I? I would learn whether they were gram positive or gram negative and what shape the cells were. And that's very useful information, which is why that's usually the first test we do is gram staining. But if I got a mixture of these organisms that arrived in the lab, let's say they were all part of a patient's sample, some poor patient was infected with all four of these things. If that arrived in the lab and I had four things on my smear and I gram stained it, I'd still get some information. I would be able to see I had both gram positive and gram negatives in there, but it starts to become less useful when I see that I have two different organisms that are gram negative rods. I have two organisms that are gram positive. Do you see what I'm saying? It's important to gram stain. That's an important piece of information when you're trying to identify something. But you can't stop there. You cannot look at a gram stain smear and identify something. You just can't. You need more information. And in this particular group of organisms, it turns out that oxygen category is going to be a very useful piece of information for us. You wouldn't know that, but I know that, which is why I chose those four for us to look at today. So I took some thioglycolate broth tubes and I inoculated one tube for each organism. I also took some nutrient agar plates. I didn't use the nutrient agar plates though to prove that they were alive. Remember, I did that before the experiment started. But instead, I made some smears on the nutrient agar plates so that I could put some into the Brewers jar. So for the nutrient agar plates, for each organism, I tried to grow it in a Brewers jar and I tried to grow it in room air. That will give us a little information too. And finally, I did a catalase test. For each one of the organisms, I did a catalase test in order to see whether or not they had the catalase enzyme. So let's first think about what we're doing and what we might see. Make some predictions, in other words. So let's think first about those nutrient agar plates. Remember, some of them I'm going to grow in the Brewers jar and some of them I'm going to grow in room air. So looking at the 5 oxygen categories, what should be able to grow on a nutrient agar plate that is incubated in room air, in oxygen? Think about the 5 categories. Obligate aerobes, yes, of course, because they require oxygen. So yeah, I expect them to grow. Facultative anaerobes, yes, they'll grow in oxygen. They prefer oxygen. Aero tolerance, micro aerophiles. Both of those organisms will grow in the presence of oxygen. One of them requires oxygen. A micro aerophile requires oxygen. The aero tolerant will tolerate oxygen. But for both of those, the organisms are only going to grow poorly for us in room air, because remember, both of those categories prefer a low oxygen environment. You will grow a micro aerophile, you will grow an aero tolerant in room air on an agar plate, but it's not going to grow well. And remember, when I say that something is only going to be able to grow poorly, what I'm saying is the colonies are going to be really small. They're not going to be as big as I would expect them to be. There's only one category missing in that list, and that's the obligate anaerobe. An obligate anaerobe cannot grow in room air. Oxygen is toxic to that organism. So I will expect nothing to grow. Now, let me just stop here and say that if you need to go, because it's just about 11 o'clock, I understand. If you can hang out for a few more minutes, that's great. If you need to go, just remember that you'll want to go through the lab video once it's posted to the playlist, at least the last 20 minutes or so of it, so that you see what the experiment showed us, okay? Okay. Now, what about the organisms growing on nutrient agar in the Brewers jar? Remember, what a Brewers jar does is it takes all of the oxygen away. So here's where our obligate anaerobe will grow. I would expect to see growth on the nutrient agar plate for the obligate anaerobe. But that's not the only thing that will grow in a Brewer's jar. Remember, we have 2 categories that will grow when there's no oxygen around. Aero tolerant, again, it'll grow, but it's going to grow poorly because it prefers an environment with low oxygen. So it's going to grow for us because it doesn't use oxygen to make ATP. It just prefers to have a little around. And the facultative anaerobe is also going to grow because this is the one that will prefer oxygen, but will grow with or without oxygen. Now, with regards to the Catalase test, remember, we expect to see a positive Catalase test if we're dealing with an organism that has oxygen inside of it in order to do its metabolic processes. An obligate aerobe requires oxygen. So yes, it's going to have oxygen inside of it. So it's going to need catalase to break down any oxygen byproducts. We'll also see a positive catalase with a facultative aerobe because even though they don't require oxygen, they prefer to grow in oxygen. And finally, the micro aerophile should have a positive catalase test because they require a low level of oxygen to make ATP. The other two categories, the obligate anaerobe and the aero-tolerant anaerobe, should have a negative catalase test. And that's because they just don't have oxygen around them. That's the obligate. And the aero-tolerant can't make hydrogen peroxide. So these two should have a negative catalase test. Remember, we're going to see patterns of growth according to which category these organisms belong to. It's going to look similar to this drawing with one big exception. The drawing shows the cells all separated out in this part of the tube, all separated out in this part of the tube. We're not going to see that. We're going to see strings growing, hanging down like tendrils into the bottom parts of the tube. Okay? All right, now we're ready to go through our results. So, with regards to the nutrient agar plates, on nutrient agar in room air, so we incubated it in room air, for Clostridium Sporogenes, no growth. Nothing grew on that plate. For Enterococcus Fecalis, it grew, but it grew very poorly, very small colonies. For E. coli, it grew great, good-sized, normal colonies. For Alkalogenese Fecalis, it grew, and it grew great, nice, good-sized, normal colonies. Now, what about the Brewers Jar? Clostridium Sporogenes grew great in the Brewers Jar. That's a big clue, right? It didn't grow in room air at all, and it grew great in the Brewers Jar. Enterococcus Fecalis, it grew just fine in the Brewers Jar. Nice, good-sized, normal colonies. E. coli grew in the Brewers Jar, but it grew poorly. Compare that to how well it grew in the air. And finally, Alkalogenese didn't grow at all in the Brewer's Jar. That's a big clue for that one as well. This is the slide we looked at just a minute ago. Remember, we made predictions about what can grow in room air and what can grow in a Brewer's Jar. So compare those predictions to these results and see what you come up with in terms of what you're thinking so far. We said that in room air, anything that doesn't grow in room air, anything that does not grow in room air should be an obligate anaerobe. They are the only category that doesn't grow in room air. Does that make sense? Clostridium Sporogenes, at least right now, looks like it might be an obligate anaerobe. It grew great in the Brewers jar. It didn't grow at all in room air. Okay, let's take a look at the tubes. So this is the thioglycolate media, the semi-solid thioglycolate media. We did not use the kind that has an indicator dye. We just used the regular kind that's the same color throughout the tube. Now take a look at this tube. These results were actually a little bit disappointing because we didn't get a lot of growth in here. But if you look down here at the very bottom, you see that white clump right there? Those are cells. I don't know why we didn't get more growth. I can't tell you why, but we did get growth. This clump of whitish colored material down here, these are bacterial cells. And I think you'll agree they are growing at the bottom of this tube. There is nothing growing in the rest of the tube. Remember, when you look at a broth, if the broth is clear, if I could hold up a piece of paper with words on it and read the words through the broth, that's a broth where there's no cells growing. When cells grow in a broth, the broth becomes cloudy or even opaque. I won't be able to read words through it. So this little clump down at the bottom, that's in the anoxic zone where there's no oxygen. That makes sense if we're thinking that Clostridium Sporogenes is an obligate anaerobe. So far, so good. Here's our Enterococcus Fecalis. And this is what I mean by tendrils, folks. Take a look at this tube. It almost looks like a sea creature or something, doesn't it? Now, notice that there's a lot of growth right here. There's a clump. That's what this is. These are millions and millions of cells that are all clumped together. Most of the growth in here is right here. But I also see these tendrils hanging down. There's a big heavy one right there. Here's another tendril growing. Here's another tendril growing. What happens is, because there's agar in this, there's a little bit of agar. Not a lot, but a little. The cells are going to tend to clump together as they divide and grow, rather than being evenly dispersed all the way through the broth. The important thing to note is where the tendril is growing. I have cells growing all the way through this tube. Most of the cells, though, are right here. Most of the cells are in the lower half of the oxic zone. Now let's take a look back at our drawings for a moment. See if one of these tubes looks like that to you. Do you see one of these tubes where cells grow all the way through, except for the very top of the tube? And where cells grow the most in the lower half of the oxic zone. One of these drawings matches that description, doesn't it? No growth right at the top. Lots of growth in the lower half of the oxic zone, most growth, in fact. And then additional growth through the rest of the tube. Any ideas? Which one do you think this which category do you think this organism belongs to? Best growth in the lower half of the oxic zone, but some growth throughout the entire tube. What do you think? Mackenzie's got it. Carrie's got it. Aerotolerance. That's an aerotolerant. That's an organism that doesn't use oxygen to make its ATP, but it tolerates it. It's not toxic at all. And in fact, it likes a little bit of oxygen in its environment. It likes a low level of oxygen in its environment. So it grows really well in the bottom half of the oxic zone, even though it's not using that oxygen to make ATP. It'll grow all the way through the tube. It doesn't care about oxygen. It tolerates it. It doesn't care about it. It doesn't need it. But it grows best in that bottom part of the oxic zone. And that's exactly what we can see in that tube. Here's our E. coli. Now I have two tubes for us to look at for E. coli because E. coli didn't read the textbook apparently. And it didn't grow quite as well as I might have liked it to. So I have two different test tubes to show you that two different students made. I want you to look at this one on the right first. This organism grew best in the oxic zone. That's what you're seeing here. But it also grew in tendrils in the rest of the tube. And that's what you're seeing here. Now, these tendrils aren't very long yet, but for some reason, this culture didn't grow very fast. So you can see some tendrils starting to come down. And if we put this tube back in the incubator, these would have gone all the way to the bottom. The reason I wanted to show you both pictures is there's a tendril here. It's right here. And it's growing all the way to the bottom. It's just very faint. So this is how I want you to interpret these two tubes. We have growth all the way through the oxic zone, and we have tendril growth through the rest of the tube. So unlike the last tube, we have growth in the upper part of the oxic zone too. This one over here, it looks like maybe there's no growth up top, but that's just an artifact of the way I took the picture, I'm afraid. It really did grow all the way to the top. So heavy growth in the oxic zone, some growth in the rest of the tube, some growth down in tendrils in the rest of the tube. What do you think E. coli is? Most growth up in the oxic zone, top and bottom. Some growth in the rest of the tube as tendrils. Which category do you think E. coli belongs to? This is an organism that actually prefers to use oxygen if it's available. It'll do aerobic cellular respiration if it can. But if there's no oxygen around, no problem. It'll just switch over and do another metabolic pathway because it's very adaptable. And in fact, most bacteria are in this category. That's right. Yun's got it. Carrie's got it. Facultative. This is a facultative anaerobe. E. coli is a facultative anaerobe. It prefers to do aerobic cellular respiration if there's oxygen available for it. But if there is none, it's okay. It'll just switch over to either anaerobic cellular respiration or it'll switch over to fermentation. Good, very good. This is the drawing for a facultative. It can grow all the way through the oxic zone. It grows heavily in the oxic zone because it prefers to do oxygen requiring metabolism. But if there's no oxygen around, it's happy to grow without it as well. So let's draw some conclusions. Clostridium Sporogenes, this was probably the easiest one. This one had no growth in room air and good healthy growth in the Brewers jar. This is a classic obligate anaerobe. We saw growth in the deep anoxic region of the thioglycolate tube. This organism finds oxygen toxic. This organism must have an oxygen-free environment or it will not grow. Alkalogenese Fecalis, well, This one kind of had the opposite. It grew in room air. It didn't grow in the brewer's jar. And it grew only in the oxic zone of the thioglycolate tube. It grew only in the oxic zone of the thioglycolate tube. Take a look right here. Here's Alkalogenese. See that? There's no tendrils down here. There's nothing growing down here. It's all right here. Alkalogenese Fecalis is an obligate aerobe. This is an organism that must have oxygen or it will not survive. And just like we saw in the drawing, it can only grow up in the top of the tube, in the oxic zone. So we have an obligate anaerobe. We have an obligate aerobe. Then things get a little more confusing. Enterococcus Fecalis. This is the one that grew in room air. It grew in the brewer's jar, but poorly. And it grew in the lower oxic zone, as well as the rest of the tube. Best growth in the lower half of the oxic zone. This was the one you told me was aerotolerant. Aerotolerant organisms do not use oxygen to make ATP, but they tolerate it. And strangely, they prefer to have a little bit of oxygen in their environment. They grow best when there's a little bit of oxygen in their environment. Finally, we have our friend E. coli. E. coli grew great in room air. It grew poorly in the brewer's jar. It grew all the way through the thioglycolate tube, but it was best in the oxic zone. E. coli is a facultative anaerobe. Good. Oxygen categories. Thioglycolate tells us which category an organism belongs to. Thioglycolate broth is not the same as nutrient broth or any other kind of broth. It's a very particular testing broth. We don't use thioglycolate just to grow something. We use thioglycolate to figure out what oxygen category it belongs to. So it's not the kind of broth that we would use as a culture media. It's a broth that we would use because we're trying to figure out what pattern it grows in and what category it belongs to. So Mackenzie, the Clostridium Sporogenes was the one where we got a little tiny bit growing way at the bottom of the tube. So it only grew where there was no oxygen. It grew in the brewer's jar, also no oxygen. So Clostridium is an obligate anaerobe. Clostridium Sporogenes is an organism where oxygen is toxic to it. It has to be in an oxygen-free environment or it won't grow. I'll finish up today just with a little example for you that might help you remember that. You've probably heard of tetanus, and tetanus is caused by another Clostridium called Clostridium tetani. What do you know about tetanus? What kind of an injury tends to produce tetanus? Type it into chat if you know. What kind of an injury somebody gets where the physician has to think, uh-oh, that puts them at risk for tetanus. There's something very specific about these injuries that makes a physician think tetanus. Marilyn, you're really close. Wires and nails. How do you step on a wire or step on a nail Does it have to scratch you, or rusty, old, sitting in the soil for a while? How do you injure yourself with that old rusty nail? What is the physician looking for? Is it that you scratched yourself with the nail or something else? It turns out that what makes a physician think about Clostridium is a deep puncture wound. Stepping on the nail so that it goes up into your foot. Stepping on the wire or putting your hand on the wire so that it penetrates. Deep wounds. Clostridium tetani is an anaerobe. It wants an oxygen-free environment. It needs to go deep into your tissue in order to grow. Clostridium tetani is not going to happen from a scrape. It's not going to happen from a cut. It has to be a puncture. And it has to be a pretty good puncture, like probably an inch in. So anytime that a patient comes in with a deep puncture, especially on something out in the environment, because Clostridium lives in the soil too, the physician will say, when was the last time you had a tetanus shot? Because that's what that organism likes. It likes the deepest, darkest, unoxygenated or less oxygenated regions of the body. Little bit of trivia for you. All right, everybody, thanks for hanging out late. Thanks for your patience today. We will see you on Wednesday. You have a lecture exam, but we're still meeting for lab. Okay? So we'll see you on Wednesday. Enjoy the rest of the day.
Thank you.
Take care.